What is a vaccine? Today's topic is vaccine. Any idea? Have all of you administered COVID-19 vaccine? Then without knowing what is vaccine, you administered COVID-19 vaccine. Okay, if I'll ask you, what is a disease? How will you define disease? NCRT. Okay, so once NCRT have to be gone through. But what do you understand by disease? Try to define. You can't say no idea. Okay. What is a disease? Have you heard AMR, antimicrobial resistance? A very important thing for exam also. Antimicrobial resistance. Suna hai? Okay. Have you heard antibiotic? Antibiotic. What is it? Antibiotic. हाँ, very good. A medicine? Okay, then what is disease? Fight against disease means any disease. Okay, sometimes you might have heard, like doctors used to say, in very common, like antibiotic ka dose बढ़ाना पड़ेगा. If some disease is not cured, doctor used to say, like the dose of antibiotics to be raised. ऐसा कुछ सुना है? हाँ, mg कुछ increase करना पड़ेगा. Okay. To see, first understand the basics. We have discussed in biology that is a scientific study to understand the living. We have understood the process in living, that cell, tissue, organ system functioning is organism. We started in a great detail to study this study scientifically, and over a period of time, a course of bachelor developed in medicine and bachelor in surgery related to this detailed study. We have come to know in the process that how an organism functions. That is, organism functions with organ system. So, a detailed study started in the functioning of organ system. And in the functioning, we have come to know that as the days goes on, as the growth happens in the organism, over a period of time, there comes some abnormalities to the organism. Such abnormalities affects the normal functioning of human being. For example, like the normal body temperature gets affected. Such abnormalities is called as fever. Sometimes the normal functioning gets affected in the respiratory tract. It is called as cold. Is clear? So these are the things that are the abnormalities. But there are some abnormalities when happened even poses the life-threatening issues. For example, when there happens abnormalities called cancer, is a life-threatening. Clear? So, in this process, the normal functioning is scientifically studied. And in the normal functioning, I'm just using one word, there are some abnormalities which comes in the life cycle, and this abnormalities is called as disease. So I asked you, what is disease? Very simple. I have said definition must be very simple and informative. Any such abnormalities that comes to an organism in the form of mild or severe symptoms is called as disease. Clear? So, science started in a great detail about disease. And science have come to know that What is the responsible cause for disease? So science have come to know that the responsible cause is microbes. Because you know one thing, it is the microbes, mainly the bacteria, which functions inside our body. And we have discussed at a great length what is biotechnology. Studying bio to develop technology, clear? We have discussed that how we have come to know that there is one enzyme that converts RNA into DNA. There is an enzyme that cuts the DNA, clear? So we have come to know many such many such microbes, for example, bacteria, enzymes, which are having their own function in the regulatory process of human body. And at the same time, as they functions in the regulatory process, so what happens? Sometimes when there happens the abnormalities in their functioning, it causes the disease. So one thing is clear, that are the microbes responsible for regular normal functioning also, and some abnormalities to microbes results in abnormal, that is called as disease. Clear? Now it is studied that what all types of microbes are responsible for this. Main is the bacteria. Main factor is the bacteria, most important microorganism. Then fungi, then protozoa. Is clear? Mainly these three are having the responsible factor behind the abnormalities to human body. Clear? Now, so according to all these three, when all these three in any such way infects, I'm using a new word, infects, all these three in any such way infects the normal functioning, that infection is scientifically called as disease. Clear? So as you know, any living body, say for example a living body like human, a living body like bacteria, any living body is having its structural and functional unit as cell. And we have discussed what is the cell. In very simple, I can say one thing. Cell is a chemical. Cell is a chemical having carbon, hydrogen, oxygen, nitrogen. Simply we can say, we have seen that how the functioning of this chemical results into various process. So as the microbes or living is a chemical functioning, so the abnormalities that comes due to this is also can be cured by the use of some chemicals. Clear? And you know, this chemical is known as medicine. Is clear? So under this course, there started detailed study of medicine. So very simple. What is medicine? A chemical formula to cure a disease. Simple. Now, they started a detailed study. For example, fever. What happens? Temperature rises. Clear? So, what chemical or what microbe responsible for this? So, accordingly, a chemical is developed to make normal the fever, called as medicine. Now, out of all these three, it has been found that the main role is of bacteria. Clear? Main role is of bacteria. Till the starting of 20th century, when our science was evolving, MBBS was evolving, the main cause for disease was considered as bacteria. Clear? I'll be coming to this line again. So main cause is bacteria. So mainly the medicine that started development to kill bacteria effect is called as antibiotic. Is clear? Medicine to kill fungal infection, antifungal. Medicine to kill protozoa infection, anti-protozoa. And medicine to kill bacterial infection is antibiotic. Is clear? So now there started detailed study of medicine under this course, and there have started development of antibiotics. So I asked you, what is antibiotic? Very simple. A medicine developed to kill the effects of bacteria is called as antibiotic, because bacteria is the main responsible factor behind a disease. And biotechnology even have explained that how bacteria is the responsible factor for disease. We have discussed in biotechnology that bacteria is having a multiplication tendency. So whenever something foreign comes to bacteria, bacteria multiplies and it spreads to the entire body. That we used to call as infection, that is disease. Is clear? Now, antibiotics developed under this course, and there have started use of many antibiotics to treat various diseases. Now, over a period of time, now just understand one case study. Over a period of time, antibiotic use is started. Here, but just take a case. There are some factors. For example, people residing in very humid climate, people residing in very cold climate. There are different type of climates. Point number one. Point number two, people are having some different type of dietary behavior. Clear? Third, people are having weak immune system, and some people are having the intake of medicine is more and more. Clear? I have used some 4-5 causes. That is, say for example, a people residing in a particular area. Clear? What does this mean? That in a particular area, according to weather climatic pattern, according to dietary pattern, according to various causes, there are particular type of disease prevalent in some area. For example, sometimes in common language we say seasonal fever. You can understand what is seasonal fever? A main cause for absent in class. Seasonal हो गया है. Sometimes I asked to someone, why absent in class for a long? They said, seasonal हो गया है. Even doctors have started saying, seasonal हो गया है. What does this mean? A sudden change in season. Sometimes very hot, suddenly rain. Clear? So what happens? When such sudden thing happens, microbes become active, resulting in spread of infection, and infection spreads from one to another. That we used to call in simple terms as seasonal. Is clear? So what does this mean? That there are some factors much more prevalent in a common type of disease. Correct? Factors I have taken example of many. Again I am repeating this line. There are much more factors due to which a particular type of disease is prevalent in one area. Correct? Second point. In that area, a particular type of disease means a particular type of bacteria is getting active. Correct? Third point. So if say to an area, a particular type of disease is more prevalent, so what will happen? Doctors in the area will prescribe the particular type of medicine to the area. Is clear? So just take a case. A geographical area A, a doctor is prescribing an antibiotic B. Clear? So what does this mean? That whenever the season will come, again the same. I have taken one example, season. Second example, say for example, someone is having a habit of taking more and more medicine. Is clear? So there are many such cases. So what happened in all such cases? That in a particular area or a particular type of person have become habitual of taking a particular type of antibiotic. Correct? Process is repeating year on year. What does this mean? That we have discussed in phage therapy. Recall phage, we have discussed that how a bacterial effect is memorized by one phage. Clear? So what does this mean? that when biotechnology was introduced, such thing is started studying in a great detail. That what happens, that sometimes it is noticed that many medicine given to person, whatever high dose is given, becomes inactive to work, became a matter of serious research. That what exactly the reason behind this is clear. So what I have said, antibiotic is developed under MBBS to find out that if particular type of this is the disorder, disease, this can be given. But over a period of time, there are so many factors I have said, it is found that antibiotics failed to work, whatever high dose given to a person residing in that area. Clear? There started a detailed study under biotechnology. And you know, biotechnology studies about bio to develop the technology. So biotechnology started studying in a great detail behavior activities of bacteria. And biotechnology introduced that what happens over a period of time, bacteria is the main interest, but entire microbes over a period of time develops a resistance. They memorized the composition of this chemical called medicine and they develop a resistance. So what happens? They have memorized that whenever we get active, a chemical is coming to kill us. So they develop resistance. Clear? So this is called antimicrobial resistance. Is clear? But our main interest where? In antibiotic resistance. Is clear? So what is antimicrobial resistance? A resistance developed by microbes over a period of time against a particular type of disease. Is clear? So WHO started the detailed study of AMR and WHO started publishing according to the area. Clear? I've taken the example of different geographical area. So WHO started coming out with class of antibiotics. Antibiotic first class, second class, third class, according to the AMR study. So just take a case. If in an area the basic antibiotics are best preferred, no need to have go to second one. So according to WHO study, pharmaceutical companies develop antibiotics, that is medicine, and according to AMR study, doctors prescribe the medicine for a particular area. Is clear? So you can understand what is the main basis of developing the medicine is the WHO AMR study. Is clear? On the basis of AMR study, there happens the development of medicines of different class. Is clear? Now, see. So far we have seen what is disease and how to treat the disease. In the outbreak of some disease, mainly polio, children were getting affected. And in the starting of 20th century, now medical science was at very challenging position to treat such disease by which children get deformities. Clear? Now, there started various research and it was found that microbes are not responsible for such. Clear? So there started a detailed study and what is being found that there is something new that have taken entry that is not the microbe, that is called as virus. Clear? Now, what the difference between microbes and virus? If I'll ask you what is virus, how will you define? Okay, or what is a virus? Definition. See, understand. Virus is something very small, micro, having a genetic content. So genetic content is also present in bacteria, But what the difference? Viruses in nature are existing like non-living materials. Non-living materials. No effect till the time it will enter into a host cell. Virus is in air, no issue. But the moment it enters into a bird, it enters into a human, what happens? What is the meaning of entry? Reaches to cell. What is cell? Structural and functional unit of life. Clear? So what happens? The moment it enters to a host cell, becomes living, gets active, and having a genetic content, both RNA and DNA type, so affects the genetic makeup of the living organism. If entered into the bird, what happens? Affects the genetic makeup of the bird cell. Clear? Enter into human, affects the genetic makeup of human genetic content. Is clear? So, what is virus? Having a very simple gene. Clear? Is a non-living thing, gets living when enter into the host cell. Is the definition of virus. Is clear? Now, so it was found that there are certain diseases caused by virus to which any class of medicine failed. Any class of medicine failed. Clear? Now coming to medicine again. What is medicine? To kill the microbes. Clear? But this microbe, nature is very different. What the difference in nature here? This microbes get living, that is its genetic content gets active when reaches to a host cell. Now, coming to basic science, recall what I said. Gene is the DNA is the special element. Who makes a special DNA? Gene. And what we have studied, that gene is everything. Gene codes for protein, and every functioning of human body happens due to genetic functioning. Think of it. Everything is happening due to gene, and someone affected the gene of host cell. That is, when the virus gets entry, the genetic makeup, say of human, gets altered. Why altered? Because a new genetic makeup have attached to the host cell. So what will happen? If the new genetic makeup now attached, so what is the work of gene? Gene codes for protein and body regulates, that is body functions. But think of it. Now there is something, a new gene, which is also coding a protein. So when which will also code a protein, there will be symptoms. And this symptom is called as disease. Clear? So now this new type of disease is called as viral disease. Clear? Now, no medicine will work. Why? Because it is affecting the genetic makeup. And what is the work of medicine? Medicine kills microbes. So we call it as antibiotic. Clear? Antifungal. Because it kills the microbe. But here, what is happening? The genetic makeup is getting altered, is getting changed. Point number one. Point number two. There started detailed study of this virus and a new branch, because by this time, biotechnology was evolving. So when biotechnology was evolving by this time, a new branch was introduced known as virology. So what is virology? Which studies in a great detail about virus. Clear? Now, what virology proposes about this virus? That virus is having a genetic content. Point number one. Point number two, what happens? This genetic content, when enters into the host cell, results into viral disease. Point number one. Point number two, this genetic content, say for example, one virus have taken entry. So this genetic content when enters, get mutated. Get mutated means Many copies it make. One entered, reaches to host cell, gets mutated. 1 lakh copies. What does this mean? 1 lakh virus now active to infect 1 lakh cells. Is clear? So there are so many things we have studied about virus under virology. So what we found, that why medicine failed, because medicine killed the microbe, and here what is affected? Gene. And that too, the virus have get mutated, lakhs of copies, we can't count. So medicine will not work to kill 1, 2, 3, 4 microbes. Is clear? So now what to do? So it is proposed that only one option: prepare the body before entry to the virus. Understand this line. So you can understand the difference between this approach and this approach. This approach is after entry, medicine is given. Here, prepare the body before entry of a virus. How to prepare? So it is proposed by virology that all these properties of virus to be studied in a great detail, and after the study, it is proposed in virology that after the study, according to the genetic content of a virus, clear? According to a genetic content of virus, as virus gets mutated, so according to genetic content of a virus, now the body is to be prepared to make learn that, see, this is type of gene, if it will enter, to make clear the immune cells about this entry. So what will happen when the immune cell will get understand that this is the type of gene that enters, then the immune cell system will start releasing antibodies. Clear? So according to the study, it is proposed that a formula to be made. Recall what I said here, a formula. What is the formula here? Chemicals. But what is the formula here? A formula on the basis of what? Study. Study of what? Virus. So, very simple. A formula is to be made where first, step 1, study the gene of virus. Step 2, on the basis of study of gene of virus, develop the antibodies. What is antibodies? Protein against infection are the antibodies. That is very simple. 2-point approach. First, make clear the immune system, that what is the genetic makeup of this virus. Clear? So take a case. For example, polio virus is studied. What does this mean? Polio virus genetic makeup is studied. Step 1. Step 2. Polio virus genetic makeup, the surface protein of polio virus is inserted in human. All human. Clear? This is the formula. With this insertion, with this injection, what happens? Human immune cells learns that this is a foreign gene. The moment it will take entry, it will start releasing antibodies. So what does this mean? That according to the virus, there have started development of the surface protein study, which is inserted into the body, and the body in return will start releasing antibodies. Clear? So what is the formula? First, virus study. Clear? Second, what in this study? The genetic content of the virus. Clear? So in the genetic content, its surface protein is injected. Its surface protein is injected. Clear? Injected where? Host cell. After the injection, what will happen? Think of it. In a living body, there comes a new surface protein, a new genetic makeup. So what will happen? Our immune system will read that this is foreign. So after this injection, what will happen? The body will start releasing antibodies. Recall, antigen and antibody in blood group. Capital A is antigen, that is surface protein on blood, and what is capital small a or b? Antibodies. So very simple. What is antigen? Surface protein. What is antibodies? Protein against infection. Clear? I can say this is infection. Why? Because this is not our gene. This is someone's foreign gene. Clear? This formula is answered, called as vaccine. Is clear? So now what is vaccine? Try to memorize. A formula under biotechnology by which a surface protein of virus is made, injected or inserted into the host cell, by which the host cell releases antibodies, that is received acquired immunity. Immunity we have natural, but this is a immunity acquired artificially, that is by the means of biotechnology. Clear? So on the basis of virology, biotechnology introduced a new industry called vaccine. Clear? Recall I have said in applications of biotechnology that we'll be discussing in a great detail about vaccine in the topic vaccine. So what I'm going to discuss here, it is to be also written in the mains question of applications of biotechnology. Is clear? Now, a new industry started. So what is the basis of development of this vaccine? You can understand, basis is virology. So there started a trend, that is virology research centers study about the virus. They collect the virus sample, they takes it to virology lab and they studies the virus. What they studies in virus? Gene, surface protein, and on this very basis there started development of this formula. Clear? So over a period of time, 5 types of formula are developed. Is clear? So very simple. What is a vaccine? A formula, a means to inject is the vaccine. Clear? First formula, that is first type of vaccine, is called as live attenuated vaccine. Try to memorize also. Live attenuated vaccine means, live means this is a vaccine where a living virus is used to inject surface protein. Clear? Directly. Most effective vaccine. A living virus is taken, surface protein is introduced and injected to the person's body. Very effective, but there is a challenge. This can't be given to children, persons having weak immune system, because what is the carrier? Carrier is a living virus. So there will be some symptoms of the carrier also, which is taking the gene of a virus. Is clear? But to few diseases like chicken pox and all, such vaccines are used. Is clear? Now, second, recall one thing we have discussed, that in biotechnology we studies about microbes and all detail. When we studied about one bacteria called Bt, what we found? That Bt bacteria releases a gene, Bt gene, and what the release from gene? Protein plus chemical substance. What is that called? Toxin. Clear? Biotechnology studied that this liquid toxin is very effective as a carrier. Clear? So toxins of bacteria were collected from the lab and they have developed a new medicine, that is vaccine called toxoid vaccine. Very simple. So what is this vaccine? Toxins of bacteria are used. Is clear? Now, most effective, but what I said, can't be given to many. So a vaccine is developed in which the dead or inactivated form of microbes are given. Just take a case. A bacteria is taken, bacteria is made dead. Clear? And simply a dead bacteria is used as a carrier. Clear? So a dead bacteria is taking what? A gene to the body. Clear? So as bacteria is dead, so bacteria will not do anything. Bacteria have just used for carrier. First case or second case, if say there is an active bacteria, we have clinically made the bacteria inactive. What does this mean? That the bacteria is inactive, so bacteria will not have its impact. Clear? Will work just only as carrier. So this is the medicine, vaccine, which can be given to any children even. Polio vaccine is of this type. You can understand what I said regarding here. So try to memorize living and here non-living, something inactivated or dead. Is clear? Now, sometimes it came to notice that there is no use because the surface protein that we are injecting is very negligible. Is very negligible in its content, 0.001 mg, very negligible. So why to use a full organism? Cut bacteria, half bacteria, half virus is used. That is not the full unit, subunit, so called as subunit vaccine. Is clear? So subunit means not the full unit. A bacteria is made cut. Is clear? And just used as a carrier. Now, the last one. You know what is antigen? Surface protein. Clear? We have antigen on our blood. We have antigen in various viral organism. Clear? It means there are so many type of antigen present in biotechnology. So mix many antigen. Mainly mix strong antigen with weak antigen. Clear? So mixing, just try to memorize in one word. Mixing is called as conjugate. Is clear? Now, so under biotechnology, a new concept, a new technology is developed that is called as vaccine. And over a period of time, this branch of biotechnology have remarkably worked well to treat various viral disease. Is clear? As I have said, 20th century, starting of 20th century, such disease became very challenging and medical science approach failed. So biotechnology vaccine industry answered to work towards this. Clear? Now, understand one thing. So 5 type of vaccine started its development and we started working to work towards any effect of any type of virus. Is clear? Now, but when this research started under biotechnology, some scientists talked on this line that, understand this line, I can say one thing, vaccine industry is completely dependent on virology. Correct? A virus is studied, study is given to vaccine industry, and vaccine industry develops 5 type of vaccine. Clear? Some scientists started talking that, think of it, if there will be a virus, because virus nature is what? Mutation. If there will be a virus that gets mutated in numbers, understand, there is a virus that gets mutated in number. Clear? But if there will be a mutation where a virus is coming to host cell of human and human is having another genetic content, clear? So if both will change into a new type of variant, then this entire industry will collapse. Understand this line? I'm repeating again. That vaccine industry is completely depend on virology study. Few scientists talked about that if say there will be a virus entered into three person. First person, strong immune system. Second person, weak immune system. Third person having cancer. What does this mean? In all the three person, human genetic content is having different effectiveness. Clear? A person having cancer, oncogene is affected. Clear? So what does this mean? That the virus, when it will enter, if will get mutated, then the virus that will come out from the person having cancer will have the impact more severe than the virus that is coming out from the person having a strong immune system. You are getting this line? Then the entire vaccine formula will fail. But this research was not progressed because human life have not encountered any type of such virus whose surface protein is similar to our cellular machinery. Is clear? As never such virus have happened in its entry in even in research, even in disease. So no work have been started to this research. But a virus have come that is called as coronavirus that placed a serious question mark to this entire concept. Clear? Because this is for the first time a human life have countered such a virus whose surface protein is similar to ours. So the moment this virus reaches to our lung cells, surface protein matches, lung cells receptor opens, virus enters and damages the entire lung cells. After damaging the lung cells, now the virus, spreads through air. So when after damaging one person, now when the virus is coming out from the person and getting spread by the respiratory tract, the virus gene is not the same which have taken the entry. While mutating in the lung cells, virus gene is also mutated. What does this mean? That now the virus which have taken the entry, when it is having the exit, it is not the same. So no vaccine will work. Then what to do? Is clear? This is the challenge made by coronavirus to the entire industry. Clear? So one thing I have said was clear. Scientists have made it very clear that if such type of thing will happen, this entire study will collapse. Because this study is based on what? A virus is having this genetic content. On this very basis, antibodies are made by the body. This is the formula of vaccine. But in this case, now the entire concept failed. So in case of COVID-19, now what to do? So the answer is one. So to which the research started. Only one answer. Entire concept failed. Only one answer. That answer is the genetic vaccine. Is clear? So now one thing became clear. And as I have said, this was introduced in research, but never this research was progressed and no company have ever worked in this type of vaccine. Because there are so many challenges, that is challenges in terms of preservation, in terms of work and all. So first I'm explaining to you in very simple, what is genetic vaccine? In vaccine, we are sending the surface protein. In such vaccine, it is something like training our immune system that see, if this is the gene, you have to release this antibody. But if gene is getting changed, you have to release this antibody. You can understand. Here, instructing and here, training. the thing is not so simple. Clear? So there started the scientific research to develop the genetic vaccine. Clear? I'll be coming to this research. So scientists have answered that to this type of vaccine, just answer is just one. But scientists have said that we have started this research now. It will take time to come to this point. Clear? So now what to do? So entire biotechnology research started somehow working to use all 5 formula to develop something for coronavirus. Clear? Now, I'll be coming to this at last. So what to do then? So entire biotechnology research in vaccine industry started working for all these 5 formula to develop somehow a vaccine for coronavirus. Clear? Now in this vaccine, now what is proposed? That leave the case, first do one thing. Take the coronavirus genetic content and do the same. Point number 1. But while doing, try to perform in such a manner that at least it will work to maximum number of people. Clear? There started vast research, and for the first time, Russian scientists claim to one success regarding this research. Clear? Now, what they claim? See. Have you heard of adenovirus? Once it was asked in prelims. And as I have said, almost every year they are asking one question from vaccine. Adenovirus. What is adenovirus? Any idea? सुना है? Okay, see. Sometimes when you say viral हो गया है, viral fever हो गया है, clear? What you mean to say? Even goes to doctor and says viral हो गया है. What you want to convey? You used to say or not? कि viral हो गया है. Clear? क्या ऐसा हो गया है जिससे लग रहा है कि वायरल हो गया है? हाँ, फीवर एंड कोल्ड, गुड. एंड व्हाई यू से वायरल हो गया है? हाँ, वेरी गुड. मीन्स ये बहुत कॉमन है. क्लियर? लगभग सबको हो गया है इस तरह के, फॉर एग्जाम्पल सीजन चेंज एंड ऑल. क्लियर? नाउ साइंटिफिकली व्हाट इज दिस वायरल हो गया है? एडिनोवायरस. Adenovirus is most common virus in human life. They're even much common that sometimes you find like someone is coming from very heat, 45-degree temperature, and suddenly drinking cold water. You can understand? So what does this mean? That the moment sudden changes happens to our body, this virus get active. Clear? But this virus does not cause serious disease. Its nature is to cause cold and fever. Clear? So what is adenovirus? Is the most common virus responsible for viral fevers, that is cold and all. Is clear? Now, for the first time, Russian scientists remarkably worked well. And as you know, the world's first COVID-19 vaccine made by Russian scientists, that is known as Sputnik. They named it after the first satellite launched by then USSR. Is clear? Now, what the concept Russian scientists introduced? They came to this point that adenovirus is present in all. So do one thing. Use adenovirus as a carrier and place surface protein of coronavirus in the adenovirus and insert to human body. Is clear? Then what will happen? Point number 1, no rejection of immune system, and this vaccine will at least work some of its work to everyone. because every human body is trained with adenovirus. Is clear? You are getting this point? Again, I am repeating. As almost every human body is having this virus, what does this mean? Our immune system is well trained with adenovirus. Clear? That how sudden impact this virus get active? So first, they decided to do one thing. Use adenovirus as carrier and put the surface protein of coronavirus on adenovirus and insert into the human body, called as COVID-19 vaccine. They named this vaccine as the responsible factor for this virus is viral fever. So they named this vaccine as viral vector vaccine. Is clear? World's first vaccine, that is a Sputnik, is made on this formula. You might have heard or even have taken Covishield. India's Covishield is made exactly on this concept, because Russian scientists afterwards, they have given this formula to many, and on this very basis India have made Covishield, the same vaccine that is of Sputnik, that is viral vector vaccine. Is clear? आगे समझ? Second, this vaccine, you can send, can't answer completely, but at least this vaccine have worked well and much more effective to the person having strong immune system. Why? Because at least our strong immune system person body now trained to release antibodies against coronavirus genetic makeup. Is clear? This research progressing. By this time, again a new concept introduced. In this new concept, this concept was implemented, that as coronavirus is very severe one, so why not to use a living virus, take a living virus and use as a carrier. This will work well. Clear? So far, this is the most effective vaccine in the world. You might have heard India's co-vaccine. Clear? Co-vaccine type vaccines worked well, but it is suggested to only give to persons having a strong immune system and all. And sometimes you might have noticed, like after the vaccine dosage, doctor said like there will be fever or something due to effect of this living virus. Clear? So this vaccine is called as whole virus vaccine. Is clear? Now, third type, which have not become popular and almost not in much more use, but one third type is also introduced, that is based on this protein subunit vaccine. Is clear? So there are mainly three type of vaccines developed for COVID-19. But one thing was clear, that it will not work with 100% efficacy. Sometimes Covaxin claimed 90%, sometimes any other vaccine claimed 60-70%. Now, the time passes, after 6-7 months, again a new wave came. In the new wave, what came to notice? Now the variant is completely new. No relation between the previous and the now. So what does this mean? That the previous vaccine will not work. So they introduced a concept of what to do then. So do one thing, take a new vaccine. The new vaccine is called as booster dose. Now just think of it. What, by all this development, biotechnology have read the same thing what was suggested by scientists, that if such type of virus will take entry, coronavirus is just one case. If such type of virus will take entry, what does this mean? That if this will be the basis, vaccine concept will fail. Today new variant, it will take time to develop vaccine formula, then by the time so many casualties. Again this formula is developed, virus have changed its variant. You can understand, it's not possible to take for one disease so many vaccine doses. So only one answer, that is the genetic vaccine. So they started work on genetic vaccine, and in this work, since 2020, a success is achieved to which Nobel Prize 2023 is awarded. Is clear? Now understand about this. You know what is gene? In simple, I can say RNA and DNA are having the genetic makeup. RNA is single strand and DNA is double strand. Clear? Now, what is introduced in this? First, understand. First, I am explaining to you scientifically what is a genetic vaccine. In a genetic vaccine, the genes is introduced by a surface protein. Gene is introduced. So what happens? The gene now when comes to our body, see, what is the work of vaccine? It releases antibodies. But now understand the difference between genetic vaccine and other vaccine. Genetic vaccine means a genetic content is introduced into our body. Clear? So what will happen? In our body, there is a new genetic content, point number one. So this genetic content will first develop antigen in our body. What is antigen? Surface protein. First step, not antibodies. First step is antigen. Clear? Now there is a new surface protein that is coming in our body. To counter this antigen, now our immune system will develop antibodies. This is the genetic vaccine. There have started many such development. Few companies have claimed also, like Moderna, J-COVID, are the DNA vaccine, RNA vaccine. But the problem with this research is, so far, approximately 60 to 70% efficacy is only achieved. Clear? Many companies have come out with many formula. Such vaccines are called as RNA vaccine, DNA vaccine. But what the problem with such vaccine? That 60-70% efficacy. That only 60-70% it works, and it fails to work on many changing variants. Is clear? So they started the scientific research, and one scientific research have greatly claimed about one formula to which the Nobel Prize is received. Now work is going on that basis of Nobel Prize to develop such type of vaccine. Now we have discussed in the basic, you can recall, what is nucleoside. Nucleoside, न्यूक्लियोटाइड, कुछ याद आ रहा है? न्यूक्लियोटाइड यहां था, कुछ यहां से चले थे हम, यहां रुके थे, and I've written nucleotide. While I stopped, I said something about nucleoside. वो सब याद भी रखने हैं. Okay, notes को याद नहीं करना है. You have to, and see how bacteria is memorizing. See, and you are not memorizing. Okay, तो nucleotide क्या है? हाँ, nitrogenous bases plus sugar. Clear? Made a chain. But recall what I said. Nitrogenous bases meets with sugar. Clear? The time this meeting happens, before coming of a phosphate group made this chain stronger. But before coming to phosphate group is called nucleoside. Is clear? Phosphate group now made the chain, so called as nucleotide. Clear? Now, scientists have said one thing to which the Nobel Prize is awarded, that they have researched out that the time nucleoside forms, because recall what we have discussed, that with this process we come to understand how the genetic function starts, chromosome. Clear? So scientists have suggested one thing, that the time nucleoside gets formed, do one thing, nucleoside base modified. Clear? Modify nucleoside and use this modification as a carrier, and mRNA. What is mRNA? Messenger RNA will be used as a carrier to make available the genetic content of the virus to the body. Once again, I am repeating. In a successful research, scientists have claimed that the time nitrogenous base meets with sugar, do one thing: modify this. Before phosphate group comes, modify this molecule, and this molecule will be used as a carrier. And to this molecule, now the genetic content of the virus will be placed. And it will be used as a carrier. And who will make available this carrier? mRNA will make available this carrier. This carrier have reached. So what our body will do? Our body will release antigen. Antigen. So our body will come to, and what is this antigen? Surface protein of whom? Virus. Clear? So our body now, the releasing antigen, countering this, our body will then release antibodies. Clear? So to this formula, now the vaccine is called as mRNA vaccine, and to this formula, Nobel Prize Medicine 2023 is awarded. Very important question for prelims. Clear? Now you can understand. With this Nobel Prize, at least science and biotechnology research have received one direction, that this is the way we can work towards development of genetic vaccine. Is clear? So till this is clear to you all? Okay. Now, if I'll ask you, how many of the vaccines are approved in India for COVID-19? COVID-19 के कितने वैक्सीन इंडिया में अप्रूव्ड है? Which all vaccine you all have taken? Who all have taken co-vaccine? Okay, others Covishield. Okay. There have come a report. Okay. उस रिपोर्ट से डरना नहीं है. It's clear. They have come some report that while the process was in hurry, so might be some side effect to human population. Okay, but not to have any fear to this. Okay, so anyway, how many vaccines are approved in India? Okay, name all three. नहीं वो नहीं, booster dose नहीं. My question is, how many vaccines are approved in India? Total 8. And have you heard there is one nasal vaccine also introduced? But for that vaccine also, it is not like anyone can administer. For that also nasal vaccine, one has to go to the vaccination center. Okay. So total 8 vaccine is approved in India. One nasal vaccine is approved, and recently there is a success, and Government of India is running a program called Mission COVID Suraksha mainly for this. Clear? There have come some success. A company Zydus Cadila have claimed to develop a Zy-COVID. They have claimed it as world's first DNA vaccine. But I have said before this Nobel Prize that whatever be the claim of the company, the challenge is 60-70% maximum. It is not working effectively. That is why scientists have introduced to the companies that you can use this formula for mRNA vaccines for COVID-19. It's clear. And recently, one Gemcovac, just few months before, a Gemcovac was in use. Again is claimed of one genetic vaccine. Is clear? And many such vaccines are also been approved also, but is not in the use. Approval means that such vaccines are in the trial phase. Is clear? So, regarding prelims, any such vaccine, if comes in use, now you are in a position to understand what is vaccine, what is COVID-19 vaccine, what are the challenges in COVID-19 vaccine. So, if any such news comes from the vaccine topic, it is to be updated. very much important for both prelims and mains. Is clear? Okay. So now it is clear.
So by mutation process, person to person, the virus gets severe.
Mainly severe. Their virus gets even virus become mild also. But our interest is in what? Severe cases. That is why recently India have performed one of the largest genomic surveillance just few days before, which was even while this is presented. Now genomic data is given to labs to study about the effects of genetic mutation, that how virus affects this type of population, how virus affects this type of people. So on this all basis, now the future development is going on. Is clear? Now you can understand in such research, supercomputers and AI are having a remarkable presence. That is why you have written in supercomputer Param Pravega, a supercomputer installed at IISc Bangalore. अभी ये अच्छे से भूल गए हो गए, okay? Because ICT topic we have discussed long ago. From Param Pravega, anyone can say its capacity? When the time will come when you will memorize, schedule is not yet released. So all of you have to appear by this prelims or not? इस prelims में किसे किसे appear होना है? 25 वाले में? Okay, means you have to launch by 26. That is why going by this slow process. See, the basic concept always to be in mind. Is clear? And the rest of the thing is a matter of revision. That is before your exam. But basic concept always to be in mind. Okay? You have written in Param Pravega, largest capacity HPC developed in India for scientific research. 3.3 petaflop. Clear? Main objective of this is genomic surveillance. Think of it. If such capacity supercomputer will be using, then only we can progress towards vaccine development. Is clear? Because it's not possible always to use a vaccine, then perform a clinical trial, then come to a formula. So with one particular composition, now many scientific studies performed by which a vaccine is made. Is clear?
Sir, what is the difference in giving the vaccine after or before the enter of virus?
See, after the virus, the vaccine dose is not having any meaning. You can understand, coronavirus outbreak was sudden, and as I have said at very first, that is why you might have heard there came a study that coronavirus is man-made. Still, and this has become very much controversial also among many Western countries and China. Still, this is not rejected by WHO, this theory, because it is for the first time a virus surface protein imitates our lung cell protein. Clear? So there is no meaning of taking vaccine after. But as there was no option, so this was made. Because vaccine means what? Before. And as accordingly, virology performs almost all possible spread of virus. Clear? Even takes the virus from air, goes to the lab, perform the study, present this study to vaccine industry, that this type of viral disease affects children, affects this type of people. So accordingly, vaccine industry developed the vaccine. Is clear? So vaccine means before. COVID-19, sudden outbreak, that is why such work was made. Clear? Otherwise, there are various vaccine formula that is at work. For example, you might have heard sometimes coronavirus is written as SARS, COVID virus. Clear? SARS. This is called severe acute respiratory syndrome. Clear? There are so many such, pneumonia and all are also one type of such disease. Clear? But why? All such SARS viruses like coronavirus was not the case. because it gets mutated and change its variant. That is why the formula get failed. Otherwise, to SARS virus vaccine, there are so many vaccines present in the market, but all such vaccine why failed? Because the nature entry is same respiratory system, but the nature is completely different. That is why the entire concept failed. Is clear?
Sir, recently the outbreak of HMV virus.
हाँ, HMPV. हाँ, what all study you have gone through with HMPV, or have come about the something like दूसरा wave कोरोना की तरह? Have you gone through with such so many studies? So many such reports were becoming viral. To the sources you much more refer, social networking sites. दूसरा कोरोना वायरस HMPV. Okay. So see, HMPV is like other viruses. Clear? What the thing most challenging was, notice that affecting children in the long run. So now labs with HMPV virus genetic study now have declared that now we are well prepared to fight with this virus. Clear? So you can understand. HMPV type such virus, chimpanzee virus and all such are sudden outbreak. Clear? Now virology after COVID-19 have expanded its study also and going to study much more virus also. Is clear? Say for example like from pigs, pigs are also having many such virus that transmits to human easily. So many such virus are studied. So virology have expanded its study to develop about the vaccine. Is clear? But to coronavirus type virus, only answer is genetic vaccine. Is clear? Then write about this. Under the topic vaccine, write first paragraph, disease. Right. Is a abnormalities that comes in the functioning of, in the normal functioning of human body, which ranges from mild to severe symptoms, mild to severe symptoms, and sometimes also life threatening. Next point, change the line and write. In the scientific study, it is found that microbes like bacteria, fungus, protozoa are mainly responsible for such abnormalities. So, to kill such abnormalities, so to kill such microbes, a chemical formula is prepared which is termed as medicine, which is termed as medicine. Next point, to kill bacteria, medicine is termed as antibiotics. To kill fungi, medicine is termed as antifungal medicine, and to kill protozoa, anti-protozoa medicine. Change the paragraph. Over a period of time, it was found in the scientific study that due to several factors responsible like like weather, climatic pattern, dietary habits, more intake of medicines, etc., the microbes develops resistance. Microbes develop resistance and makes the medicine ineffective to work. Ineffective to work. Such resistance develops by microbes which developed by microbes which makes the medicine ineffective is termed as AMR. Underline, this is very important definition. Antimicrobial resistance. Antimicrobial resistance. Change the paragraph. AMR study is conducted on a wide scale by WHO and also by various health organizations, and on this very basis the classes from lower to higher of medicines, that is antibiotics, are made. Antibiotics are made to fight against AMR. Fight against AMR. Change the paragraph. In the outbreak of disease, a microorganism happens to be non-living, called as virus, made challenging the medicinal formula. Made challenging the medicinal formula. As virus is having a genetic content and as a non-living component, exist, but the moment it enters, the moment it enters to a living host cell, gets active, gets active, and with its genetic content, with its genetic content results into mild to severe symptoms, mild to severe symptoms known as viral diseases, known as viral diseases. To treat such diseases, a medicinal formula is made to prepare the immune system before entry to the virus, before entry to the virus, by introducing by introducing a carrier which consists of surface protein of the virus, surface protein of the virus, comma, by the introduction of which to the body, by the introduction of which to the body, there starts release of antibodies, there starts the release of antibodies which works against the viral spread. This formula is known as vaccine. This formula is known as vaccine. Change the paragraph. In order to study in detail about virus, there started a separate branch known as virology under biotechnology, and on the basis of this study of virus, following 5 types of vaccines are developed. Following 5 types of vaccines are developed.